Good morning. Welcome you all to the next lecture on inorganic chemistry of life, principles and perspectives. In the previous couple of classes, I have tried to introduce the enzymes based on vanadium and based on manganese. In case of vanadium, we have seen vanadium bromoperoxidase, chloroperoxidase, haloperoxidase, and also we have looked at the inhibition of the vanadate in ATPase cycle. And on the other hand, in case of manganese case, we have looked at manganese superoxide dismutase, which is one manganese based one. Then we also looked at the manganese catalase, where there are two manganese ions are there. Then we have looked at the oxygen evolving system of the photosystem 2, which is tetranuclear manganese cluster. So, we have looked at all these three. So, for these cases we have looked at all the mechanistic aspects and more details, and the rest of the items were just covered as a information into that. Now, let us smoothly get into the next topic, that is on the iron in the biological inorganic chemistry. So, here we may be taking a bit longer time because the iron case is quite huge. There are a huge number of enzymes present in the biological systems, both based on the heme and non-heme. So, therefore, we will be spending a bit several more lectures on the iron biological inorganic chemistry. Okay, let us look at this particular slide. As you can see, the iron is very special because there is something called heme, which I have already introduced in the introductory courses classes, and then we have a non-heme too. And look at the heme proteins. They can do oxygen transport, they can do electron transport, they can do oxygenation, they can do reduction, many kinds of things, variety of oxidations, reductions, everything. Look at non-heme, where there is no heme. They can do transport, they can do electron transfer, they can do oxygenase, okay, they can do deoxygenase, they can do reductase, they can do hydrogenase, variety of functions are there. Okay. So, iron case we also have the ferritin, which is the iron storage protein, and a transferrin, which is iron transport protein. Okay. So, I think we will try to start with transport systems first, then we will go to the other cases. Okay. Oxygen transport proteins. So, oxygen transport proteins, which we are very well aware in human systems, is hemoglobin. Which we are not so commonly aware is other things like Hemerythrin Hemocyanin, not for human systems, but these are for other kinds of things. I will show you in the next slide. So, the short form for hemoglobin is used as HB, a short form for hemerythrin is called HR, a short form for hemocyanin is referred as HC. Let us look at this particular table, which tells you the various properties, various aspects of these transport proteins. As you know, hemoglobin is for the human, higher animals, hemerythrin is in vertebrates, and hemocyanin is arthropods and molluscus. So, as you can see, the hierarchy of the life changing from the higher life to somewhat intermediate, somewhat to lower kind of thing. So, these let us look at a few of the properties. There is the iron is the form of heme in the hemoglobin. In hemerythrin there is no heme, okay, and there is one iron, one oxygen in hemoglobin, two iron with one oxygen in hemerythrin, two coppers and one oxygen in hemocyanin. So, coppers, not irons. So, metal and oxy in oxy protein means oxygenated one. In case of hemoglobin it is iron 2 plus, in case of hemerythrin is iron 3 plus, in case of hemocyanin it is copper 2 plus. Deoxy form, that means when there is no oxygen bound, then there is again Fe2 plus in the hemoglobin and Fe2 plus in case of hemerythrin and copper plus in hemocyanin. You can see that there is a 1 electron difference in this. One should think of where does this 1 electron go. So, this 1 electron must go into the oxygen, the O2 that is bound, whereas here there is no change in the number of electrons in the oxidation state, therefore, O2 does not accept any electrons. Straight away one can derive such information from here. Okay, so because iron 2, here's iron 3, iron 2, obviously 1 electron difference, copper 2, copper 1. And you know that there are 2 ions, so 2 cases, 2 into 1 electron here. There are 2 coppers, 2 into 1 electron here. So that means O2 is not O2 in these things, possibly O2 with some 2 electrons or something of that kind. We will come to all those details a bit while, just hold on your breath to understand more details. Now come to the form of the color aspects of it. Oxy form, in case of blood we know very well blood is red, and hemerythrin case burgundy, and in case of hemocyanin blue. So these are called the blue blood animals. The arthropods and molluscus are referred as the blue blood animals. We are red blood animals. Color of deoxy form, red purple in case of hemoglobin, and almost colorless in case of hemerythrin, and almost colorless in case of hemocyanin. The reason is this is in copper 1 and this here is copper 2, but due to some other reason the color is lost here and the color is still there. Okay, so let us look at what kind of a binding course that these have. We mentioned 1 iron, 1 oxygen. In case of hemoglobin, you see that this whole thing is heme, and this is the iron center which is connected through the 4 nitrogens of the porphyrin, and there is one more nitrogen coming from histidine, which is called proximal histidine, which is coming from the protein chain, side chain of the protein of the hemoglobin. So, globin chain, and on this side you have a 6th coordination where the oxygen is bonded. Now, in case of hemerythrin, the oxoform is bonded to the O2 is not like O2 like this. Here it is already, what is this? This is like peroxo or hydroperoxo, and you know that the peroxo and hydroperoxo are formed from O2 after 2 electrons reduction. And that is what I showed you on the previous slide just by looking at the iron oxidation state in the oxy form versus the deoxy form. Very easy. And of course, the 2 ions are bonded by 3 histidines, 2 histidines respectively, and these are bridged by the carboxylates. So, we will come to more details later on. Okay, and in case of hemocyanin, the 2 coppers, again O2, this O2 is O2 minus. So, there are again 2 electrons, copper 1 becomes copper 2, therefore 2 electrons, that will become this one. So, the way the oxygen is present in human blood is more or less unperturbed O2 or least perturbed O2, and whereas in case of these hemerythrin and hemocyanin, the one of the bond is almost reduced from the double bond of the O2. So, which means when it releases, it should gain back that. So, therefore, there is quite a huge mechanism involved in these delivery of the oxygen, which are not very well understood. So, we will not go into more details, but we will compare only the features, but we will go into more details of this of the hemoglobin. Okay, look at this particular structure shown over here on the right side, ribbons, you know, alpha helical structures, etc. Not ribbons, this kind of a helical structures, and there are some random coils. Very little of the beta sheet will be there, and in the center there is a core here. There is a core, protein formed a core. In this core, you have the heme, that is the porphyrin and center metal, iron, and this histidine. Histidine is coming from this particular helix. This particular thing is myoglobin. It has only 1 unit, and its molecular weight is 12.4 kilodalton. Now, on the other hand, if you connect 4 such units, I will show in the next slide how they are connected. The 4 such units, then it becomes hemoglobin. So, myoglobin is 1 unit, hemoglobin is 4 such units. So, 4 into 12.4, 49.6, around 50 kilodalton is the molecular weight. So, but what happens when the 4 of these are connected? The resultant protein, which is called hemoglobin, and it is not a myoglobin, and there are changes in its properties too, as we will see in the next one. So, let us see that how a hemoglobin looks like. Now, hemoglobin looks like, you can see first 1 heme here, that yellow portion, 1 more heme here, 1 more heme here, 1 more heme here. That means The 4 such units, one on this side, one on the back side, one on the front, one on the rear. So, totally 4 very symmetrically connected, it is called quaternary structure, and 4 myoglobins are joined together to form a hemoglobin. But the myoglobin, now let me tell you, myoglobin means myo is muscle, so it is present in the muscle, and it is involved, the myoglobin is involved in mostly in the storage of the oxygen, whereas the hemoglobin, hemoglobin, hemo referring to the blood, so hemoglobin is involved in transport. So, when we have one protein unit, it acts like a storage. When you have 4 such units are joined together, it acts like a transport protein. This looks like very interesting, isn't it? How come one unit of the protein is just a storage and 4 such units joined together? There is something called cooperativity. That means these 4 units which are joined together do not behave as an independent or individual units, rather they jointly work as a cooperative unit. So, it is this cooperativity which brings in the transport property for hemoglobin and storage property for the myoglobin. So, the storage versus transport we are going to discuss and debate in the next several slides. Why is that? One is the transport storage alone, other is the transport. Okay. So, whatever I said till now, I have summarized in the form of structures on this particular slide. This whole thing is your myoglobin and this is your heme portion. Okay. So, you can see the myoglobin and this is the hemoglobin, one such unit, one more and one more, 4 units are there. And if you expand any one of this unit and you can see this, so this one and this one looks exactly alike, no difference at all almost, but each of these is interconnected. So, therefore, you have this is what we were trying to say, that one unit is not good for transport, is good for storage. When you connect the 4 of these units, then it is the protein has a transport phenomena and it shifts from the storage to the transport kind of a system. Now, first of all, look at some features. If some protein has to be a nice transport protein for oxygen, what kind of qualities should it have? What kind of qualities should it have? Number 1, it should bind, the protein must bind to O2, number 1. And number 2 is the O2 should not get perturbed at all. That means O2 should not lose its bonding double bond character, or O2 should not receive electrons to itself. Okay. And there are some additional things are also there. Okay. So, let us look at this one. So, you need to have oxygen binding to the ion center, that is one of the quality, and then the protein should stabilize this complex in this form, not in the reduced form. And in the binding pocket of the O2 in the protein, it should not allow the oxygen to get either 1 electron reduced, 2 electron reduced, or whatever it is there. It should not happen. That means O2 should not get activated, or O2 should not get reduced by electrons. Either of these terms are one and the same. Okay. They are all one and the same. And one more thing which we do not know generally, but when you think of the chemistry, then we will certainly know. Iron 2 ions, you know very well. You take a iron 2 salt, let us say ferrous chloride, and dissolve in water and just go out for a cup of tea and come back. What you will see? You will see quite dark brownish color, sometimes even a precipitate. If your tea time is too long, you might even see a precipitate too, or your water is bit of a alkaline, also you can see that. So, there is an oxidation, there could be hydrolysis, all these kinds are possible. Similarly, when an iron 2 in hemoglobin or heme binds to oxygen, why should it not get oxidized? Yes, it would get oxidized. So, therefore, iron 2 P refers to porphyrin. If we add O2, temporarily obviously it will make iron 3 with a kind of a peroxo, because 2 electrons will go, and over a period of time it will lose one of the oxygens, etc. There is a mechanism not shown over here, and that will form iron oxo iron 3. Iron 3 oxo iron 3. This is called myoxodimer. Once this is formed, there is no reversible. So, that means your oxygen is fully activated, O2 to peroxokine to the myoxo. So, that means O2 is completely destroyed. So, such kind of thing should not happen. So, that means your hemoglobin should, or even myoglobin for that matter, to store O2 in its real form, you require to maintain the bond order and should not. So, therefore, the protein should make a complex with O2, should not reduce the thing, and iron 2 plus should be protected so that there is no myoxodimer is formed. One more difference is required for the protein, because the substrate for this protein is O2, not the CO, and so therefore, there should be recognition of CO versus O2 by the protein. So, CO is a diatomic molecule, O2 is again diatomic molecule, and how does the protein know whether it is a CO or O2? The only difference is only a very small difference. How does that happen? So, if it does not recognize, then always the carbon monoxide will bind and that will passivate your protein. So, therefore, this is an important event. We will come back to that. How does the protein, whether it is a myoglobin or hemoglobin, differentiate CO versus O2, that we are going to look at in a while. Now, for a while let us focus on two aspects. One is no oxygen bound case, other is oxygen bound case. So, it is called oxy form, it is called deoxy form. The left side is the deoxy form, right side is the oxy form, and take this thick line as the perpendicular view of the porphyrin. So, that means this is a porphyrin plane is like in line, the iron is bit above that. So, it is showing around 0.6 angstrom above the cavity of the heme. Now, if you put the oxygen, this is still above, but not as much, it is about 0.2 angstrom. So, almost about 0.4 angstrom drop has happened from the iron from its plane. So, the iron is here, plane is here. So, and then it comes closer. So, when you have oxygen is bound, then the iron comes closer. When oxygen is not bound, it is farther away. So, let us look at in the deoxy form. Of course, in both the cases, iron 2 is a D6 system, and you can see that you have a high spin system. And on the other hand, once you make the oxygen ligand, then you form the low spin complex, and because of that, iron is able to come and sit in this. That I will explain in the next slide, why in the oxy form iron is more fit into the cavity of the heme, and whereas it is not so much fit in the heme. Just wait for a while, and the next slide I will be explaining that. Or if you think in the meanwhile it is already good. Okay, clue for this is, I tell you, I give a clue. There is a high spin iron center, there is a low spin iron center, both are iron 2. That is the clue. Now you can understand why it is going into the thing. So, I will explain, just unravel that in a while, but you are always welcome to think of on that lines as well. Hemoglobin without the bound oxygen. The Oxy-Hemoglobin is with the bound oxygen, and it has a lower absorption 660 nanometer, Deoxy has a 940 nanometer, and this is the difference that is used for measurement of the amount of oxygen present in the patient's blood by using the pulse oximeter. In most of the hospitals and labs where you go for pathological labs, where they check your blood samples for oxygen purpose, they will do that. Now, you understand that the D6 configuration is in high spin in case of deoxy and low spin in case of oxy, and I told you to think why the iron is coming closer to the plane in case of oxy and not so in that. Now, you see the high spin ion, you know that how do you define a size of an ion. So, we define a size of the ion or even an atom to the extent from nucleus to the extent to which the electron density is spread. So, therefore, it is the spread of the electron density that you need to look at that. So, in case of high spin, the electrons are filled not only in the ground in the other excited level, higher energy levels too. So, therefore, the size of the ion is bit growing or longer. So, therefore, it is roughly about 0.8 angstroms in case of deoxy. Now, the moment you put the oxygen, and then oxygen once you put the iron 2 goes into the low spin, and once it goes to the low spin, all the 6 electrons are paired into the ground state, T2G, it is a 6 coordinated system, octahedral system. So, T2G6, therefore, the electron density is somewhat compressed, therefore, the size is basically compressed. So, almost from 0.8 to 0.6 is the kind of a change which is about 25 to 30% change in this. So, you know that from the coordination chemistry principles that what we studied is that we know that the oxo-ligands are not very strong field ligands, they are rather weak field ligands, and they generally encourage the system in the case of as a high spin complexes. But in case of hemoglobin or the myoglobin, when the oxygen is bound as an additional ligand, but it prefers the low spin. So, it is not because of ligand field strength. When this is bound, there are some conformational changes come, and these conformational changes indeed favor the low spin configuration of the electrons in the D system rather than the high spin. It is that part. It is not the oxygen has become suddenly a strong field ligand. No, it is not true that. So, therefore, what we know, the coordination chemistry is correct, but there are several things which we do not know, because in the protein chemistry, the protein conformation, protein structures are also important along with the inorganic chemistry aspects of it. So, together, so here I hope you understand why what happens when the oxygen is bound to the iron size, and why, and how does it get fitted into this. I hope you understood all that. Okay. So, therefore, in the deoxy-hemoglobin it is too large, and in the oxy-hemoglobin it is smaller and goes closer to that. So, and we already have seen in earlier, the protein that is surrounding this heme does not allow the oxidation of the iron 2 plus to iron 3 plus. It does not allow iron oxo iron, which is called mu-oxo dimer formation. All of these are prevented, and it also does not allow the electrons to flow into the O2 because of the kind of redox potential that we have for the ion center in the hemoglobin and myoglobin systems. Okay, one another aspect that I talked to you is that this enzyme must recognize O2 and over that of the CO. It is very, very important. Okay, so for this, why should it? Because if you look at the cells, and if you see, the cells are always having CO being generated from the dead cells, etc., which is about 1% of the CO is always present in cells all the time. So, that means O2 must bind to the hemoglobin in presence of the 1% of the CO. That means the enzyme must differentiate the CO versus O2 at that particular concentration of CO even. If not, what will happen? CO will bind. If CO binds much better, stronger, by about 30 to 50 fold stronger, and irreversible. Stronger and irreversible. And we know the reasons of this back bonding between the metal and the ligand, all those kinds of things come into picture, which you must have studied in very simple the inorganic bonding studies, coordination chemistry bonding studies as well. As compared to the O2, so therefore, that is one thing. Now, we will come to that point just in a while, but let us see now. So, when it binds to O2, you see that this is known from the structures of the small molecule as well, even from the protein, that the FeOO is a bent structure, is around 120 degrees or so. And such a kind of structure is stabilized by two things. There is a kind of a push from this side, and there is a kind of attraction from this side, histidine, which is called the distal histidine, where there is a hydrogen bond, it stabilizes. On the other hand, when the CO binds like a linear FeCO, and this is 180 degrees, and this has no stabilizing power into this. So, therefore, the binding nature of these is different. You see that this is the deoxy. Of course, in the resting state there will be water will be replaced by O2, and then when O2 comes here, this is what binds. This is the stabilizing force. Now, if you look at, I am sure you must have heard several cases where there is an accident, a fire, people obviously die. You know the main reason for the people die, not because of they are burned, but because they are being choked with the smoke. So, that is more dangerous than even the fire, because people will run away and they can save themselves, but whereas when the smoke is spread, even if you run, then smoke haunts you, and then the smoke you breathe the smoke, and the smoke has got CO, most of it, and the CO concentration in your lungs becomes so huge, then your hemoglobin will get saturated with the CO, and once it is saturated with the CO, no reversible, and you die. And that is mainly the reason why most of the deaths are because of the CO smoke reason, not because of even the burns. Of course, I am not saying there are no deaths or the burns. What I am saying is the maximum things always, because when the fire is there, obviously firefighters will come and try to extinguish the fire, but the smoke is not easily extinguished so easily as the fire. Fire gets extinguished immediately, the smoke goes longer. So, therefore, people before they can escape out of the danger, probably they get caught into the danger of this one. So that the higher at that stage, can this protein differentiate between the O2 and CO? No, because there is a concentration shift, huge concentration of CO, even that this stability whatever you have is overcome by that. But within the cell, yes, where there is 1% of CO is there, it is always there. Okay, now let us try to look at, so we have looked at the various properties that such a proteins should should exhibit. Now look at one particular aspect of this. Suppose if a transport protein binds to oxygen very strongly, what level of strength of oxygen binding is important? Those proteins which are strong in binding will have a problem in releasing, okay? And you see that when they are strong in binding, they will go something like this, which is called the hyperbolic kind of a thing. Now, if take some transport protein, there is some transport protein which is strong, not strong in binding, but inefficient in binding, and somewhat efficient in the uploading kind of thing, then you would find the hyperbolic curve of this kind. Okay, so there is a lot of difference. Now here, if you take a transport protein is both efficient in binding as well as transporting, both binding and transporting, then it will not be neither the hyperbolic of this type nor of this type, but of this type. What is this type called? This is called sigmoidal one. So, what is the myoglobin? The red one is myoglobin here, red one is myoglobin, red one is myoglobin. So, myoglobin is always going in the hyperbolic. Therefore, myoglobin is not meant for the transport, but it is just meant for the storage of this. Okay? And let us try to see the same thing in case of the hemoglobin. Now, look at the myoglobin once again here, and this is what? This is the partial pressure O2 versus the to what extent the oxygen is oxygenated, protein is oxygenated. So, oxygen saturation, see MB is the myoglobin is going like this, absolute hyperbolic, and forget about this HB, look at this HB and here, and you see a sigmoidal. So, look at on the right side, this is a sigmoidal plot. So, in the sigmoidal plot, what you have? You have a weak binding in this region and you have a strong binding in this region. That means the protein has one kind of a conformational structure here and the protein has a different conformational structure. That means protein is going from weak binding, one conformational structure, to strong binding to another conformational structure, and there is a transition. So, that means if a protein has the qualities to have a weak as well as to have a quality of the strong, then it will be able to function both pick up as well as the transport the oxygen very efficiently. So, it can switch from the weak binding to the strong binding, and I will continue with this in the next class. So, I am trying to explain you the oxygen storage and release of these difference between the myoglobin and hemoglobin. So, in the next class I will continue and try to clarify all the things associated with the mechanistic aspects of the oxygen transport in hemoglobin, and which is not so in the myoglobin part of it. Thank you very much.